Thank you, Mark. So it's a pleasure to represent not only Mark, Helen, but also all of the other computational biologists in the audience who've not only produced all the methods that we're using over the next few years, but in the few years ahead are going to contribute to developing new methods that are going to be so necessary for the success of the Lifetime Initiative. So I think you've already understood from the talks we've heard this morning, and indeed more generally, that computational methods are becoming increasingly central to biology. Indeed, it's hard to distinguish to separate out computational methods now from modern biology. And in the context of the Lifetime proposal, there are different layers at which computational methods are going to be applied. We could think of it at the level of processing, the types of beautiful data that have been described this morning, so from single-cell genomics technologies, from imaging technologies, and also something we didn't hear so much about this morning, but I think is going to be important, the inter-individual aspect, incorporating genetic variation into this, and then how these different layers of biological information can be intersected together with epidemiological data, disease status, and then applied together with these new and very exciting techniques that Amos alluded to this morning from artificial intelligence and machine learning to ideally predict disease outcome. And also we'd like to go beyond in this project, I think, disease outcome, predicting it, and actually doing something that we could understand the mechanisms that underpin these different outcomes. So what I'm going to do over the next few minutes is really just a prelude to the presentations you're going to hear over the next couple of hours. I'm going to briefly mention some examples of how computational biology has helped our understanding and interpretation of these different types of data. And where appropriate, I'm going to outline some of the open challenges that I think we're going to have to face and overcome if this project is going to be successful over the next 10 years or so. So it's certainly true that disruptive technologies drive biology. We heard this morning about the huge advances in single-cell approaches. But in order to enable these technologies, in order for them to be used, we need to develop appropriate computational methods. This is perhaps most obviously been exemplified of what we heard this morning in the development of statistical approaches that allow us to move from analyzing bulk measurements of RNA, where we average across thousands of cells, to computational approaches that allow us to analyze single-cell genomics data, both at the sequencing level and also in the context of imaging. These approaches can take various different routes. So this is from a review that Ollie Stiegel put together, and this is an application in the context of single-cell RNA sequencing, where we want to try and understand some of the different sources of variation in the data. So here what we have is a differentiation process from a naive T cell to an activated Th2 cell, and the observed expression signal that we get is over here on the right-hand side, because it's a combination not only of this active process, but also a variety of other unobserved factors, or often unobserved factors, just such as cell cycle stage, whether the cell is stressed and beginning to undergo apoptosis, and you can imagine a whole variety of other factors. So one of the key challenges in the field is to try and disentangle these different sources of variation so that we can understand the sources of variation that are attributable to, say, this differentiation process and separate them out from cell cycle. So there's been a variety of linear models developed for this, employing approaches from factor analysis in order to try and dissect these different sources of variability and also to give cleaner data sets that we could then use in downstream analyses. As well as the sort of more low-level modeling and understanding different sources of variation, there's of course the key challenge of visualizing this data. So this is work from Fabian Theis's lab, so maybe Fabian is going to talk about it in his presentation immediately hereafter. But this is one of a variety of different approaches for visualizing single-cell RNA sequencing data. Amos referred to these earlier as the dreaded clouds of points, these with the red dots. And they are difficult to interpret, and I think one of the challenges we're going to face in the next decade, especially as we try and apply these methods and have them used in a more medical context, is having very interpretable outputs. At the moment, some of these approaches, they do look a bit like hairballs. They're not necessarily very easy to interpret. And also, there are a lot of different ways that you can slice and dice these data. You can plot them in a lot of different ways, and it can change the way that they are interpreted quite a lot. So we need to think very carefully about developing effective and intuitive displays of complex data, perhaps borrowing from other fields. You could think, for example, about, say, Google Earth and how you're able to zoom in and out so very beautifully there to local areas, to a global view. We probably need to think about developing similar tools in the context of single-cell genomics data, both in normal development and in the context of disease. As well as the sequencing technologies, there's also, of course, been huge developments in imaging. Here there are similar low-level challenges where one has to segment images, identify specific features, whether they are at the subcellular level or at a different scale of resolution. And I think one of the key challenges here at a project that is going to generate data at a variety of different scales is how we stitch them all together. We really could do with coming up with a coordinate framework where we integrate data at the tissue level down to information at the subcellular level. How these different aspects all stitch together is going to be absolutely essential for us to develop the sort of coherent models of normal development and disease that are going to be necessary for the success of lifetime. So this sort of establishment of a coordinate framework, and here I really do think that there's a lot of work that could be done, especially linking the different scales. So from a computational perspective, I think this is a very exciting challenge, and here at the top are just two examples. One where we have a subcellular resolution visualization of a particular system. Here, this is from the lab of Jan Ellenberg, and then a higher level visualization of at the tissue level, and this is from Lucas Pelkman's lab, I believe. Once we have all of these different types of data, we've processed them, we've segmented them, we've identified relevant features, deciphered different sources of variation. Of course, ultimately, we would like to stitch them all together. There are varieties of different ways that one could think about doing this. This is an example of a multi-view factor analysis model where different views correspond to different technologies. So this is motivated by data generated by Wolf Reich's lab, where Wolf's developed this technology that allows you to analyze RNA, DNA methylation, and chromatin accessibility from the same cell. And what we want to do here is to identify factors that are shared across the different views and factors that are specific to individual views and how they interplay with one another. One can imagine extending this sort of analysis to a much wider variety of different technologies. And I think that these sorts of approaches are going to be absolutely key for facilitating biological insight as well. So it doesn't have to be factor analysis. One can imagine other approaches from machine learning being used in this context, but we really need to get going as a field in developing and thinking about these methods, I think. Having said that there are still challenges, it's important to say already very substantial progress. Nikolaus and Geneviève earlier mentioned the very close links between lifetime and the Human Cell Atlas. And for those of you who are not aware, which I suspect is a very small number, the goal of the Human Cell Atlas is to define this expression profile, or profile of all of the cell types within the human body, both at the molecular level, but also at the spatial level. So they would like to come up with a periodic table, effectively, of cell types. And using the sorts of computational approaches that have been developed by people in the room, there's already been a lot of progress here. So this is work from a few labs that are all part of this consortium, I believe. So where the computational analysis at the top was done by the lab of Sarah Teichmann, and at the bottom by Fabian. And here the goal was to decipher different cell types in the lung. So although there is work to do, we have as a field already made very good progress, and there's a lot of opportunities. These opportunities come in a variety of different flavors. I mentioned earlier that genetic variation is one area where, or inter-individual variation is one area where there hasn't, maybe perhaps primarily due to cost, been a lot of single-cell omics data generated to date. There's just one example here from the lab of Luda Franca, where they looked at immune cell populations and then effectively across multiple individuals with single-cell RNA sequencing, they clustered these into different cell types and then identified cell type-specific QTLs. Now, that was a kind of a first stab at this problem. I think that there's going to be a lot more work in this area where we try and incorporate genetic variation here, both to uncover cell type-specific QTLs, cell type variants that are associated with chromatin status, et cetera, and also from a computational perspective, we need to be able to develop linear mixed effects models that are going to properly model this type of data, analogous to the sort of approaches that have been applied in GWAS studies to try and decipher and to get in an unambiguous way the different sources of variation. Of course, all of this focus is really on normal. We're also going to, over the next 10 years, really begin to see the application of single-cell omics and imaging technologies in the context of disease. I'm not going to dwell on this too much because we heard some beautiful examples in Amos's presentation earlier. But suffice it to say that we're going to have to think carefully about integrating and propagating error from these measurements at the top into these downstream approaches. There is a lot of noise, there's a lot of variability in some of these measurements, and we know that when we analyze them from a statistical perspective, but very often we just plug in raw numbers into these downstream approaches, and as Amos mentioned himself, garbage in, garbage out. if you don't process this data well at the top, then you're just not going to have useful models, you're not going to have useful predictions coming through at the end. So we need to think about how we propagate some of the sources of error at the top into these models down at the bottom here. Not only that, we also have to think about extending existing models, existing causal reasoning, causal modeling approaches, in order to try and get some insight here as we further develop these approaches from AI and ML and apply them in this field. I would say that one specific challenge, and this is particularly due to a pan-European project, is to ensure that the methods we develop are going to be applicable across all of the member states, all of the countries that participate. The example that I think is perhaps most complicated is where we think about integrating data from clinical records. Here, there's differences, of course, not only in language, but there's also differences even within a country in how such information is stored. And if we want to really apply the methods we develop across the whole of the consortium, we need to think about methods from natural language processing, other techniques that are going to be able to translate, literally in this case, translate information from multiple languages so that we can really integrate information and from sensible downstream analysis. And I think this is going to be quite important if we're going to have a successful long-term project here that's really of value to all of the European citizens. As well as that, and perhaps it's a little bit drier, but actually it's absolutely critical, is ensuring appropriate data sharing and management is put in place. The data ecosystem is incredibly complicated in terms of the coherence of metadata. This project is going to involve data from a variety of different countries. We're going to need consistency in metadata, file formats, etc. The legislative landscape around GDPR is increasingly complicated. We need to make sure that we are at the cutting edge of that and we apply appropriate approaches for ensuring the safeguarding of our data. And that's going to be necessary both just for ensuring public trust, but also we need these interfaces to ensure that the data types talk to one another. So the bottom here, this is an example of an imaging analysis of early embryo development. This is single-cell RNA sequencing of the same stages of early development here in the mouse. We need a way of linking these data sets in the store so that for the user, there's an ability of saying, well, actually, this is from the same stages of development. What can we learn by stitching these two types of data together? So with that, I'll close and leave the floor to the individual speakers who are going to go into more detail on specific aspects of their own research. But I would just say that this is a tremendously exciting time in the field of computational biology. Quantitative methods really are beginning to have an impact. And I think if we do this properly, of course, taking into account the data storage, processing, we're going to have a tremendously successful period and a really exciting period in terms of our impact upon human health, which is really the purpose of this consortium. And with that, I'd like to thank you for your attention, and I'm happy to take questions if there are any. Thank you.